All right, so let's continue our conversation about DNA, and we're going to discuss DNA replication. This is where you have strands of DNA that are going to be replicating exact copies of each other. Normally, DNA replication happens during the interphase in a eukaryotic cell, so as it goes through its cell cycle, and this all is going to take place within the nucleus. What I'm going to draw is I'm going to draw the DNA unwound, and we're going to have it open, and I'll explain why in a minute. So let me just draw what they call fork here. So this is going to be our replication fork. And this is going to be the sugar phosphate backbone. So in a previous video, I talked about what makes up DNA. So we have our sugar phosphate backbone. And right now I'm drawing the nitrogenous bases. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. They are going to be base pairs, meaning if there is an A here, there's going to be a T here. They pair up. And if there's going to be a G here, there's going to be a C here. Now, what does that mean? The nitrogenous bases, it's going to be adenine, there's 4 of them, thymine, cytosine, and guanine. Okay. All right. In DNA replication, you do have a couple of factors that are key players. So we have a couple of enzymes that are key players. We have helicase, DNA polymerase, primase, and ligase. And they all have a very particular job. Let's start off with helicase. So helicase is going to come in and it's going to break those hydrogen bonds. Now, helicase only breaks the bonds during DNA replication. That's going to be important when we talk about protein synthesis. So DNA replication, helicase is involved. Done. All right, so now we need primase. So primase is an enzyme that comes in and puts down just a very small strip of RNA, and that allows your DNA polymerase to then know where to bind. So this normally happens at the origin. So this is going to be our DNA polymerase. It's going to bind here, and it's going to start putting down DNA. This is what we call our leading strand. And this is going to go from 3' to 5', putting down DNA in a 5' to 3' direction. The lagging strand is a little bit more complicated. Okay. So you also have a primer that's going to be coming in. So let's say we have a primer here. Okay, and that's from primase. And then DNA polymerase binds. Okay, and it's going to go and put in DNA in this direction. So it's going to start making these little fragments. This is going to be called an Okazaki fragment. You're going to have, as the helicase is unwinding, you're going to have a bunch of these little fragments that are going to be made, and those will then need to be bound together by ligase. Okay, so that's a very brief overview of DNA replication. After all this happens, you do end up getting a strand of original DNA, and a strand of new DNA. And they call this semi-conservative. Okay, any questions? Let me know.